Hello everyone, welcome to Pharma Thoughts. Episode 1, Drugs and Introduction. So the main purpose of starting this Pharma Thoughts channel is for knowledge sharing. We hope this will be useful for both the freshers and experienced personnel in the pharma industry. So I once again welcome you all to this channel and for the very first episode, Drugs and Introduction. So basically initially we will be covering some of the basic topics and then we will move on to more technical subjects. Agenda for this presentation is what is a drug, sources of a drug, drug discovery and development, a brief overview, differences between bulk drug and formulations. So to start with, what is a drug? The general misconception among the normal people who are not into the pharma industry is drugs are something bad. They just generally view drugs as the opioid substances or the addictive substances, whereas who work in pharma industry know that drug means medicine. So for the purpose of this presentation and future presentations, whenever we call a drug, we mean medicine. So what is a drug? What can be called a medicine and what cannot be called a medicine? So we need to know the definition, because um, only then we will be able to classify the materials whether they are drugs or they are not the drugs. So to help us, some the regulators have provided a proper definition for the drug. So let us see some of the definitions. To start with, as per the US FDA, that is United States Food and Drug Administration, a drug is defined as a substance recognized by an official pharmacopoeia or formulary. In USA, the official pharmacopoeia is USP, United States Pharmacopoeia. So we can understand that a substance that is recognized or mentioned in the USP is called a drug. A substance, also another definition is a substance intended for use in the diagnosis, cure, mitigation, treatment or prevention of a disease. This is also called a drug. So if a substance, it can help in diagnosing or curing or mitigating or in the treatment or preventing a disease, then that substance is called a drug. Another definition is a substance other than food intended to affect the structure or any function of the body. If any substance which can affect the structure or any function of the body, then it is also called a drug. So here biological products are also included in the same definition, are generally covered by same laws and regulations, but only difference is manufacturing process. Difference exists in manufacturing process. So as per this, we can see there are four definitions, four sentences provided, out of which if you see the first one and the fourth one, and these two are like for legality purposes, whereas to understand the real meaning of a drug, these two sentences, the second and third sentence helps us. It's very clear from the second sentence that if a substance is helping are fighting against a disease, either helping us in diagnosis, cure, mitigation, treatment, or it is totally preventing a disease, then it is called a drug. The third definition is, the third line is, a substance other than food intended to affect the structure or any function of the body, it is also called a drug. Here I got a doubt, or normally we can get a doubt. We can question this definition stating that yes, potassium cyanide or any other chemical or diethylene glycol or certain other chemicals that may harm us, that may also like affect the structure or function of my body. But can they be called as drugs? May not be. We generally don't call them as drugs. We call them as poisons. Though drugs are some chemicals and poisons are also some chemicals. We don't call poisons as drugs. So, for this purpose, we have second definition as per WHO, that is World Health Organization, who define the drug as pharmaceutical product, and the definition of that is any preparation for human or veterinary use that is intended to modify or explore physiological systems or pathological states for the benefit of the recipient. So, what they say is a preparation for either for human or animal use and which is intended to modify or explore the physiological condition or pathological states for the benefit of the recipient. If any preparation in either human or in animal, if it can help uh, in the physiological system or pathological state for the benefit of the patient, the recipient is patient, then it is called a drug. So if we combine this and the third statement of the USFDA definition, then we can say that a substance intended to affect the structure or any function of the body for the benefit of the recipient, then we call it as a drug. So now we understand, like, this is my interpretation of the definitions. If you differ with this, you can always thoroughly recheck, you can think upon it. So these are called as drugs. Let me say one example. Like, we normally use turmeric. We know the turmeric in Indian foods. Obviously, in all the Indian foods we use, most of the times we use turmeric. So if you have a scratch, if you have a small wound, if you are bleeding because of a scratch or a small wound, normally if you are at home, immediately your mother rushes and she applies turmeric. This is a common experience for most of the Indian personnel, in the Indian households. So no, when in a normal scenario, we all eat turmeric as part of our curries and as part of our dishes, whereas uh, in certain situations, like when there is a bruise, we apply it to the spot to either prevent the sepsis or prevent the bacterial infection or so. So what shall we call this turmeric? Shall we call it a food or shall we call it a drug? Just apply your logic and think about it based on these definitions. So this is food for thought. Then, sources of a drug. Where can we obtain a drug? How many different sources that we get a drug from? So drug can be obtained from any of the source, from plant source, animal source, or any of the sources that we have mentioned here. Here I have given certain sources of drug and examples from each source. So let us say, from plant sources, we can either use leaves, stem, bark, fruits, roots, seeds, etc. Any of this can be used to prepare in a drug. And examples are, from cinchona bark we get quinine, which is a drug used as anti-malarial drug. And ginger rhizome, we get gingerol, which is a carminative agent. Carminative agent is nothing but that helps us preventing or reducing the gas in the stomach, alimentary canal. So gastric problems we use it against. And animal sources, certain glands, livers, blood, stomach, pancreas, etc. are obtained from animal sources. And pancreas of cow or pork is used to produce insulin, which we all know we will be using it for anti-diabetic. Generally in India we call it as sugar. So for sugar, reducing the sugar or diabetes, we use insulin. And stomach of cow, we use pepsin. Sorry. From the stomach of cow, we get pepsin. It's a digestive hormone. And sometimes drugs also can be obtained from mineral or earth sources, and these can be either metallic or non-metallic. Suppose, for example, metallic sources, we get ferrous sulfate, which is used against anemia. We know what is ferrous, it's iron. So when there is hemoglobin, less hemoglobin, then we use ferrous sulfate to treat anemia. And for non-metallic sources, hydrogen peroxide, we use it as an antiseptic. We all know, we might have some or other time have experienced usage of anti hydrogen peroxide, like when there is a wound or bruise. If you go to hospital, they first clean the bruise with hydrogen peroxide. So this hydrogen peroxide, once they pour it on the wound, we apply white foam and then they clean it. So but have you ever seen some doctor prescribe hydrogen peroxide for you to drink? No, we never drink or we never take hydrogen peroxide as an injection. But still, it is called a drug. How? So, again, go back to the initial definition. A drug, any substance that helps in diagnosis, cure, mitigation, prevention. So, this particular hydrogen peroxide helps in mitigating the intensity of the uh, infection. So that is the reason, based on that category, we can call hydrogen peroxide as a drug. And microbiological sources. Apart from the above, even drugs can be obtained from microbiological sources such as fungi, bacteria, molds, etc. And these are one of the best drugs. From fungi, Penicillium notatum, we get a drug called penicillin G, and penicillins are the wonder drugs. These are the best antibiotics which have saved lakhs of lives, lakhs of people from dying. And from Bacillus subtilis, we obtain a drug called bacitracin. Also, we obtain drugs from semi-synthetic sources. This is nothing but alteration of chemical structure. Example of such semi-synthetic drug is ampicillin. It's a penicillin antibiotic which is used against bacterial infections. So ampicillin is a semi-synthetic drug, and the key starting material for ampicillin is 6-APA, 6-aminopenicillinic acid. So to prepare ampicillin, we need 6-aminopenicillinic acid. This 6-APA is normally previously used to produce from fungi, and they used to ferment using the fermentation process or microbiological source. So we obtain 6-APA, from which the 6-APA is chemically altered to obtain ampicillin. There are multi variety of um, penicillins that are produced using 6-APA, such as ampicillin, dicloxacillin, cloxacillin, flucloxacillin, amoxicillin, etc. So these are called semi-synthetic penicillins. A wide range of semi-synthetic penicillins are produced from this raw material. So why we call it semi-synthetic? Again, based on the process. So 6-APA till 6-APA production, it is fermentation process, whereas microbial source, from then onwards, it's a chemical synthesis. So because of this dual nature, we call it as semi-synthetic sources. And synthetic sources, so it is purely by altering the chemical structure of drug. So we take basic chemicals and we alter the chemicals through chemical reactions to obtain one final chemical molecule which is used as a drug. Example for synthetic sources are paracetamol, which is used as an analgesic, ether and chloroform, which are used as volatile anesthetics. And apart from this, we also obtain drugs by genetic engineering using recombinant DNA technology or gene splicing. Examples for such drugs are insulin and hepatitis B vaccines. Apart from the above, even we, the human source, also can be uh, uh, humans also can be a source of drugs. Human sources include urine, kidney cells, stem cells, etc. Most of us might have come across recently, like once the baby is born, the stem cells from the baby are being collected so that they can be used in future diseases. So stem cells, etc., uh, can also be used to obtain drugs. For example, from the pregnant woman's urine, we obtain HCG, which will be used for treatment of infertility. So these are certain sources. So always we remember the definition of a drug. Like not all the plants or not all the roots, not all the animal glands will yield as drugs. So in order to classify one substance as a drug, it should meet the criteria that we have discussed previously. So here we are going to see the inception to completion drug discovery and development. How a new drug is discovered or created and it is marketed. What are the phases that happens in this time? So normally drug discovery is a very tedious and time consuming process. Also it's a very costly affair. So in the process of innovating a drug, normally first it starts with a drug discovery, wherein a target identification and lead discovery, lead optimization, etc. are done. And once the drug candidate selection is done, it will be proceeded to preclinical research. Preclinical research is done on animals, and then once the animal studies are completed, then it moves to clinical research. It includes phase 1, phase 2, and phase 3 studies. Again, each phase has been subdivided into phase 1A, 1B, 2A, 2B, and 3A, 3B. Once this clinical research is done on humans, then all the data that is obtained from clinical, preclinical, and other studies, this data is submitted to regulatory authorities for their review and approval. After the regulatory approval, the product is released to the market, and post-marketing surveillance will be done after it is released into the market. So typically 3 to 5 years are consumed will be taken for drug discovery, 1 to 2 years in preclinical research, and 5 to 7 years in clinical research, approximately 1 to 2 years for regulatory approval. These timelines are not hard and fast; these are approximations. And if you see, approximately 10,000 leads, if we identify, if 10,000 molecules are identified which could have the potential to become a drug, from them only 250 compounds may enter into preclinical research, may be taken for animal studies. Among these 250 compounds, successfully maybe only 10 molecules may enter into clinical research phase 1 phase 1, out of which maybe 5 molecules may enter into phase 2, and maybe by the end of regulatory approval maybe 1 or 2 molecules may withstand the regulatory approval and come into market. Even after marketing, even during post marketing surveillance, if the regulatory authorities find the risk to benefit ratio is not much, like instead of giving it instead of the drug being beneficial to the patients, if the adverse effects or side effects are more, the drug may be withdrawn from the market. So there is no guarantee that even after going through all this 10 to 16 years and doing so much research, if even if if the drug is released into market, if there are some adverse effects after marketing, then also the drug may be withdrawn. And approximately 1 to 2 billion dollars, it's a very huge amount, $1 to $2 billion may be spent on one single drug to come into the market. So this is very brief overview of drug discovery and marketing. So difference between bulk drugs and formulations. So basically, as we discussed previously, if whenever you say to your relatives or friends who are not into the pharma field, when you say that you are working in a pharma field, when immediately they revert back, oh, so you are manufacturing medicines. To them, most of the times it will be only the tablets and capsules. But if someone who is more familiar with the industry, he may ask you the second question: what is are you working in bulk drugs or formulations? So let us see what is the difference between bulk drugs and formulations. So pharma industry, manufacturing industry can be majorly classified as formulations or API industry. Previously we used to call it as bulk drug industry, but now it is either called as bulk drug industry, but most preferentially we call it as API, active pharmaceutical industry. So a formulations industry or formulations manufacturing is a place where finished pharmaceutical products, FPP, or drug products, DP, are manufactured. Please remember, finished pharmaceutical products and drug products are one and same. Only the finished pharmaceutical product is a terminology used by WHO, and drug products by US and other regulatory authorities. Whereas bulk drugs or API industry is an industry where active pharmaceutical ingredients or active ingredient or drug substances are manufactured. So active pharmaceutical ingredient and drug substance both are same. Again here the definition is in the terminology. So what is this finished pharmaceutical product and what is this API? Once we understand this definition, then we will be able to visualize, we will come to know that what is formulations and what is bulk drug industry. So let us see that. So the definitions of a drug product or finished pharmaceutical product are given here. I have taken three definitions, one as per ICH-Q7, one as per US FDA, one as per WHO, World Health Organization. So here the definitions are: the dosage form in the final immediate packaging intended for marketing. It is as per ICH-Q7. And what is a drug product? A drug product is the finished dosage form that contains a drug substance generally, but not necessarily in association with other active or inactive ingredients. This is as per USFDA. And a product that has undergone all stages of production, including packaging in its final container and labeling. An FPP may contain one or more APIs as per WHO. So from these three definitions combined together, what we get is a dosage form in the final immediate packaging intended for marketing. so that it can be directly consumed by the consumer or patient, who is nothing but patient. So you imagine, who is the consumer here? A patient. And a patient, how does a patient get the medicine? In the form of tablet, in the form of capsule, in the form of injection, in the form of syrup, in the form of ointment. So if the patient is getting in these patient is getting a medicine in these forms, and which is in its final packaging and final proper dosage, like either it should be 500 mg or 1000 mg or 25 mg or 10 mg, whatever the dosage it is. So in the right dosage, in its final packing and ready for consumption, then it is called as drug product. So this is the definition. And a drug product normally contains drug substance generally, but not necessarily contains active or other inactive ingredients. So what does this mean? Like, let us take an example. So whenever we have fever, the famous brand Crocin, we take Crocin. Crocin is a Crocin comes in the form of a tablet or capsule. So it's mostly in the form of tablet here. So when we take a Crocin tablet, it comes in a strip form. So we go to a medical shop, we take Crocin tablet, Crocin strip. So it is ready for patient's consumption. Then this is called as a drug product. So what is the drug we are taking? We are taking Crocin. Crocin is a brand name, whereas the drug actually available in Crocin is paracetamol. We also call it as acetaminophen. So this paracetamol is the drug. That is the API that is there in Crocin tablet, along with some other excipients. So here, the company, the industry which manufactures the Crocin tablets is called Formulation industry. Then what is an API? So API is paracetamol. So let us see the definition of API. Active pharmaceutical ingredient or drug substance. Again, here I have taken definitions from three different sources. One is as per ICH-Q7, one is as per US FDA, and other as per WHO. So here all the three definitions are similar. I don't say same, but they are similar. So let us go through one definition. Any substance or mixture of substances intended to be used in the manufacture of a drug product, and that when used in the production of a drug becomes an active ingredient of the drug product. Such substances are intended to furnish pharmacological activity or other direct effect in the diagnosis, cure, mitigation, treatment, or prevention of disease, or to affect the structure or function of the body. This is called an API. So in the previous example, what we have seen, a Crocin is a tablet which we take for fever, whereas a Crocin contains paracetamol. This is the main API that is used in Crocin tablet. Other things are excipients, starch or other binders, etc. So in this API, so as per the definition, an API is a substance which is used in the manufacture of a drug product. That is the tablet, and when used in the drug product, it becomes an active ingredient. So paracetamol is the active ingredient which provides the pharmacological activity, not the starch or other coloring agents or anything. They don't provide any pharmacological activity. Only paracetamol provides the pharmacological activity, and it helps in either diagnosis, cure, mitigation, treatment, or prevention of a disease. So this is called, previously we have seen, this is the definition of a drug. So an API is also a drug, but which is supposed to be used in the manufacture of a drug product. And a drug product is nothing but which is in its final dosage form and in the final packing and ready for consumption. So that is called drug product, and drug product is manufactured by formulations, whereas API is the main ingredient that is used in the manufacture of a drug product, and of when used it provides the pharmacological activity or helps in diagnosis, cure, mitigation, treatment, or prevention of disease. So the company which manufacture these drug substance or APIs is called an API industry or bulk drugs. Why it is called as bulk drug is because in an API industry we generally the manufacturing is done in huge quantities. Like the drug is manufactured in from few kgs to hundreds of kgs and tons of material. So we may manufacture from 500 grams to 1,000 kgs of paracetamol based on the required volumes. So API is a pure drug, like the final drug, which is capable of providing pharmacological activity, which will be taken by the formulator or the customer of the API manufacturer, and he further processes them into tablets or capsules or injections and dispatches it to the consumer. So here, the main difference between API manufacturing and final product drug product manufacturing is, in API manufacturing, most of the operations involve chemistry, the chemical alteration or of the structure as required. So it involves a lot of chemical reactions and other manipulations. Whereas in a drug product manufacturing or formulation industry, most of the processing is done through physical manipulations. So here, once a small raw chemical is taken and it is altered chemically through various reactions to prepare one big molecule of fine, and that molecule, that particular substance, we API industry manufactures 500 kg, which will go to formulator, where he takes the right amount of that material and adds suitable other inactive agents, excipients, and then he punches into the tablet. So in the formulation industry, physical manipulation is more, whereas in API industry, chemical alteration is more. Hope this is clear here. And these are the references that are taken in preparation of this presentation. Whoever is interested may go through these presentations or on these subjects for further data. And thank you for watching the video, and if you have any clarifications or any suggestions, please write to us at pharmathoughts1@gmail.com. It's our pleasure to answer you back. Hope you all like the video. Thank you very much. Have a nice day.